to the first. Welcome to the first AES ESA talk for year 2024. So my name is Jiafu Mao. I'm the chair of Asian Ecology section at the Ecological Society of America, which has been dedicated to advancing ecological research since 1994. So our AES goal is to enhance communication and foster collaboration among ecologists with an Asian background, studying diverse ecosystems. For more detailed information about AES ESA, please refer to our website. So as you all know, since last November, AES has been organizing AES ESA Talks, a quarterly virtual series that brings together diverse speakers from our members to field leaders, promoting collaboration and the sharing of groundbreaking research. So tonight, we are honored to welcome Professor Shulei Liu from the Institute of Geographic Sciences and Natural Resources Research at the Chinese Academy of Sciences for our second AES ESA talk. She will present thermal optimality of ecosystem carbon fluxes. Professor Shulei Liu has been with the Chinese Academy of Sciences since 2013, after earning her Ph.D. from CAS in 2004 and completing postdoctoral work at the University of Oklahoma. Specializing in global environmental change and terrestrial ecosystem functions, she has published over 200 peer-reviewed articles. In addition to her research, Professor Liu serves as an associate editor for several prestigious academic journals, including Ecology Letters and the JGR-Biogeosciences. So let's welcome Professor Shulei Liu to tonight's seminar.
Okay, thank you, Jeff. And good evening and good morning, everyone. And thank you, Jeff, and other committees of AES ESA for providing me this wonderful opportunity. And thank you all the participants for joining the meeting during the Spring Festival holiday. Today, I would like to take the opportunity to share some recent works and thinkings of my life on thermal optimality of ecosystem carbon fluxes. So the land models predict that at the 2080s, the land ecosystem will transfer from a net carbon sink to a carbon source. So the results partly due to that in the models, plants and microbial respiration are predicted to increase monotonically. Like the pictures showed in this slide, that the heterotrophic respiration and plant respiration also continually increased at the end of this century. And if we take a look at the temperature response functions in Earth system models, we can see that for those 16 land models, actually most of them use a temperature response function of monotonic increase functions like the Q10, Arrhenius, and only 2 of those models use a HAL. That means that the respiration has a peak value with the rising temperature. So that's what model does for the temperature response of ecosystem respiration. So in the real world, can ecosystem respiration increases continually with the rising temperature? Actually, theoretically, there are 3 possible response functions of respiration to temperature. The type 1 is a monotonic increased function. So that's just based on the metabolic theories. Like the enzyme reactions with the increasing the temperature. But in reality, actually, with the increasing temperature, the substrate may be limited. So there should be the type 2 response function of respiration to temperature, which means that the respiration increased with the temperature first, and then gets a plateau due to the substrate limitation or other environmental stress, especially under the higher temperature, because of the water limitation or nutrient limitation, it's most likely that the respiration may get decreased with the further increase of the temperature. So here you showed the another possible response function of respiration to temperature. That's the type 3. So what's the common response function of these 3 types of the temperature response of respiration? We analyzed those possibilities using the FluxNet dataset. We know currently we have the more than almost 1,500 site years of FluxNet datasets. We called it Flux everywhere, all of the time. Actually, for each of those flux pores, every 30 minutes, we have one site of the carbon fluxes data associated with the climate and other environment variables. So over all those flux pores, actually most of them currently have more than 10 years of continuous accumulation of the data sets. So these data sets provide an ideal platform, ideal data sets for us to test the environment or climate response of ecosystem carbon fluxes. So we use this data set to analyze how ecosystem respiration respond to the continuously increasing of the air temperature. So this work is conducted by my former Ph.D. student, Chen Weinan, and she analyzed those 212 sites ID covariance pores, which have the continuous measurement of ecosystem respiration. Actually, from those 212 sites, we found 183 sites actually show those optimum temperature of ecosystem respiration in response to temperature. Only the 29 sites showed this exponential response of respiration to temperature. So according to the previous slides shown the functions in Earth-based models, this study actually showed that in the real ecosystem, most of the ecosystem respiration not performed the monotonic increase with the temperature, but showed the widespread optimum temperature for the respiration. So we further analyzed the 9 vegetation types to see whether the optimum temperature of respiration is widespread for all the vegetation types. And actually, it clearly showed that for all those 9 vegetation types, ecosystem respiration always showed those thermal optima. So although this our study is one among the first to show the widespread of thermal optimality of ecosystem respiration, but actually at the leaf and the microbial levels, the respiration of thermal optima actually was observed and reported in previous studies. So the mechanisms underlying the optimum temperature of respiration for leaf and microbial is reported that at the leaf level, the enzyme, the deactivation and the thermodynamics of enzyme catalyzed reactions always cause the decrease of respiration at higher temperatures. For the microbial respiration, actually, the changes in microbial physiology, like the carbon use efficiency COE, and the species composition shift from the bacteria to fungi, and also the substrate limitation, all those mechanisms were reported in previous studies to explain the thermal optimality of respiration at those organisms levels. So at the ecosystem level, actually, there are some other reasons like the environmental stress. For example, the water limitation, nutrient limitation, and also the vegetation phenology. Those mechanisms together may explain why the ecosystem respiration gets decreased at a higher temperature. Because we found the thermal optima of ecosystem respiration is widely happens for all those fluxnite tars. So we further analyzed the contributing factors for the variation of TOPT of ecosystem respiration by using the ridge regression, which actually considered the collinearity among different environmental variables. We found that the temperature optima of ecosystem respiration actually mainly determined by the optimum temperature of GPP and the maximum air temperature. So for those two dominant variables, we will show the two slides following. So this figure shows the relationship between the temperature optima of ecosystem respiration with the maximum air temperature. The left panel shows that across the anticonvergence sites, while the red panel shows the relationship across the 9 vegetation types. So from both the across the sites and across vegetation type analysis, we can see that the maximum temperature can explain more than 70% of the variation of the TOPT of AR. And also, this figure shows the relationship between the optimum temperature of yarn with the optimum temperature of GPP, also across the size in the left panel and across the vegetation types in the red panels. Also, we can see that the optimum temperature of GPP predict well of the optimum temperature of AR. So you may ask that whether this widespread of optimum temperature of ecosystem respiration can be simulated by land model. So we compared the results of fluxnite versus the results from the Kibble. Kibble is one of the land models used in IPCC report. So we use the similar analysis methods as we used in previous slides. And for those 212 ID convergence sites, we analyzed the output of the cable simulations. And actually, we found that for the fluxnite, actually for those 212 sites, 189 sites showed the type 3 response function to the temperature of ecosystem respiration. But for the cable simulations, the 147 showed the type 1, that's the monotonic increase with the increasing temperature. So that's the quite different results between our observations and the cable simulations. So why the cable showed those quite different results? We further analyzed the model simulations of the cable and compare the ER results with the FluxNet observations. So this study is conducted by my PhD student Wang Song. And we found that actually compare with the FluxNet observations, the cable simulations underestimate ecosystem respiration at the lower values, but overestimated the ecosystem respiration at the high values. So the red panel shows the detail. So at the range of the ecosystem respiration that's less than 2 gram carbon per square meter per day, actually we can see the cable underestimated the values. But for the ER with above 2 gram carbon per square meter per day, actually cable overestimated the respiration. If we show the temperature response curve between these two data sources, we can clearly see that actually for the cable models, it underestimates the ecosystem respiration at a lower temperature, but overestimated the respiration at higher temperature. So what's the influence of incorporating the TOPT of ER into the models? So we compared our simulations with the incorporation of TOPT of ER, compared with those with other land models output that did not consider the TOPT of ER. So we can see clearly from this figure that incorporating of optimum temperature of ecosystem respiration will get a much lower prediction of ecosystem respiration comparing with the land model that did not consider the TOPT. So I think that's one of the reasons to get the exponential increase of the respiration at the end of century that simulated or predicted from most of the land models. Just as I showed in the first slide, that most of the Earth's models predict that at the 2080s, the land ecosystem will transfer from a night carbon sink to a south. So that's one of the possible reasons that ecosystem respiration was overestimated at higher temperatures. So this can be seen from this comparison of the result ER prediction results in the models that did not consider the thermal optimality of ecosystem respiration. So as I showed in previous slides, that the optimum temperature of GPP is one of the best explanations for the optimum temperature of ecosystem respiration. So if we want to get a better understanding for the TOPT of ER, we should analyze how the TOPT of GPP changes and what's the contributing factors for the GPP optimum temperature. So with these questions, we analyzed the eddy covers tools with the measurement of the gross primary productivity GPP. This figure just showed that among those more than 200 eddy covers sites, from the coldest sites, the US HEQ, and to the hottest sites, as shown in the US OHO, we can see that the GPP also showed the optimum temperature along with the increasing of temperature. And very interestingly, we can see that this optimum temperature of GPP increased with the site temperature. It is higher at hotter sites, but lower at the colder sites. So across all the eddy covers sites, we can see that the optimum temperature of GPP should have a good relationship with the mean annual air temperatures. To upscale our observation from the eddy covers sites, we collaborate with Dr. Shilong Piao's lab. Actually, this study is from Dr. Shilong Piao. We use the similar method to analyze the temperature response of GPP at the grid level. We can see that globally from the remote sensing data, we can see that the optimum temperature of GPP also shows high values at the hotter sites and showed the lower TOPT of GPP at the colder regions. So similar to our Eddy-Covers TOR analysis. So we found it across the sites, the optimum temperature of GPP should have a very good relationship with the air Tmax of the air temperature. However, those adaptation of the TOPT to the air temperature was strongly influenced by the water conditions. So from this figure, we can see that for the red data points, which show the dry and warm sites, we can see that the slope is 0.59. So which means that in those dry and warm areas, if the Tmax of air temperature gets a 1 degree centigrade increase and the TOPT of GPP, we also adapt to the 0.6 degrees centigrade. But for the humid areas, as shown in this green line, we can see that for each 1 degree centigrade increase of air temperature, it costs 1 degree centigrade of the increase of the TOPT. So we can call it fully adaptation in the humid or the cold areas, but the half adaptation in the dry and warm areas. So this is the across the site adaptation. For the across year, we call it thermal acclimation. We also found those close relationship between the air temperature and the TOPT of GPP. But compared with the humid areas, the dry areas also showed the lower slopes, which means that for any 1 degree centigrade increase of Tmax, the TOPT of GPP can only adapt to the 0.4 degree centigrade in dry areas. But in the humid areas, the TOPT can acclimate to 0.8 degree centigrade. So we can see that the TOPT of GPP can have those kind of a close relationship, can closely pace up. We call it thermal acclimation or adaptation to the environment air temperatures. So the question is why the TOPT of GPP can shift upward under warmer conditions. So we need to review the underlying mechanism for this thermal acclimation or thermal adaptation. So to review the underlying mechanism, especially for the biological mechanisms, we need to conduct some manipulative experiments, which provide some idea approach to review the processes, underlying processes to driving the carbon fluxes responses. So we conducted a few of the global change manipulative experiments on the Qinghai Tibet Plateau. In my lab, actually, in the past 10 years, we set up the warming and precipitation, nitrogen addition, and global change in multiple factors. Because today, we focus on the warming impact on the GPP optimum temperature. So I would only show the results of the warming experiment. So by this warming experiment was conducted in 2014, and totally we have 3 temperature treatments, including control, and we increased the soil temperature at 10 centimeters to 1.5 degrees centigrade plus, which means that we increased the soil temperature at 10 centimeters by 1.5 degrees centigrade. So another warming treatment is that we warmed the soil temperature at 10 centimeters to 2.5 degrees centigrade. So this experiment has been already run for 10 years until now. So here we show that under the warming treatment, the GPP largely increased due to 2 processes. One is the change in plant trees. Here we refer to plant height. Actually, we can see that for those 4 functional types, the bulb, legume, sedge, and grass, we can see that warming 2 warming treatments overall increased the plant height comparing with the control. And also we can see that for the higher plants, the grasses, it increased the proportion in the communities with the increasing soil temperature. But for the lowest plant functional type, bulb, actually its proportion in the community declined with the soil temperature. So it means that the species composition got shifted under the climate warming treatment. The taller species would become more and more dominant, while the lower species become more and more less dominant. So if we combine the changes in plant trees, the plant height, and also the changes in species composition, we use the community weighted height as an indicator. We can see it can explain more than 94% of the changes of the warming treatment on GPP. So which means that the GPP response to climate warming actually can be well predicted by the community-weighted plant height. So this study was conducted by my former Ph.D. student, Chen. So why the shifting of species functions from the non-grasses to the grasses can benefit the GPP? We further analyzed the temperature response functions of their photosynthesis between those 2 functional groups. So we can see that the grasses actually have a higher optimum temperature for the plant photosynthesis than the non-grasses. So here we can see that the climate warming benefits the grasses, where the grasses had a higher plant photosynthesis optimum temperature. So this will explain why the GPP had a higher temperature and warmer conditions. So we use our warming experiments. We can also explain the water regulation on the thermal adaptation or acclimation of the GPP, as I showed in previous slides. So in our warming experiments, the study sites is very unique because it cover a soil water content ranges very widely from the 8% to the 40% of the soil water content. So with this large range of soil water content, actually we found that the warming stimulates the carbon uptake, GPP, and also NEP under wetter conditions, but it's reduced the carbon uptake under very dry conditions. So this experimental results will explain our previous findings from the Edicover's sites, which showed that actually in the dry areas, the TOPT of GPP can less adapt or less acclimate to the air temperatures, but under the humid conditions, the TOPT of GPP can better adapt or better acclimate to the increasing temperatures. So to wrap up of these studies, we found a widespread emergent property of thermal optimality of ecosystem respiration, actually which challenges our traditional concept about the monotonic increase of ecosystem respiration with the rising temperature in the Earth system models. And actually we found that water regulates the thermal acclimation and the adaptation of GPP. And this variation of the thermal optimality of carbon fluxes actually can be well explained by the plant trees. In our study specifically, is the plant height. This trait-based understanding, I think, is helpful to better understand and better predict the future changes of carbon fluxes in the context of climate warming. And also from our study, that actually we have some thinkings for future studies. For example, for experiments, how to monitor the temperature response of different respiration components like the leaf respiration, root respiration, and the microbial respiration, we think is very important because ecosystem respiration just is the sum up of those different components. In order to better understand the optimal temperature of ecosystem respiration, we need to separate the different respiration components to see how their optimum temperature differed among those different components. And also we need some manipulative experiments to reveal why the respiration declined at a high temperature. Is it due to the enzyme deactivation or due to the biological regulation like the species composition shift or the other interactions among environments like the water stress or the substrate limitation and other processes? And also I think our findings have important implications for the impact of the heat waves on respiration. We previous study actually mostly focused on the heat waves impact on carbon uptake, but here we showed that the respiration also declined largely under the high temperature. So which means that this may offset the decline of the carbon uptake of the productivity, may lead to the less change in carbon sink as we previously expected. And also our study have some, inspirations for the model studies. So how to incorporate the TOPT into the model is a promising research directions. So we think we can consider these issues by those three ways. The first one is to improve the model structure, so we can modify the temperature response functions for the respiration in the models. And the other way is to parameterize the models for the temperature sensitivities. And also, our study just show a phenomenon of the widespread thermal optimality of ecosystem respiration. So in the future, we need to use the data model fusion approaches to do the attribution analysis to reveal what's the real causes for those thermal optimality. By using those attribution analysis, we can also analyze the intrinsic temperature response. So that's all my thinking about those topic of thermal optimality of ecosystem carbon fluxes. So thank you all very much for your attention, and wish you all have a happy Dragon Year. Thank you.
Nice. Yeah. Thank you so much, Shirley. Yeah, very interesting presentation. And also, yeah, congratulations on a lot of high-profile papers.
Oh, thank you.
Yeah. Okay, now let's take questions, comments from our audience. Don't be shy. So
Well,
that one want to
Sully,
go for it? Okay, cool.
okay, Sully, I can ask a question. Long time no see, and Professor New.
Yeah.
Okay.
Yeah, so happy
Okay.
to see you here.
It's a really wonderful topic, and
you.
ecosystem, thermal and optimal. And when I read your title, and sort of like you have already addressed at the end of your talk, and the different component of the ecosystem, respiration. And so I guess if we just at the very cost level, of course, you have the plant respiration and the soil microbial and respiration. So my first question is, assuming they each have a thermal optimal and or not, that can be tested. And even if they both have the thermal optimal, and will the two thermal optimal the same, or one is bigger, one is smaller? And so which one, and so what kind of mechanism? So you want to elaborate a little bit? Yeah.
Yeah, thank you very much. You raised a very important point for the different components of respiration. Actually, I showed a slide previously about the plants and the microbial respiration, and yeah, here. So actually, the phenomenon of the thermal optimality of leaf and microbial respiration actually, there's lots of the studies reported that, but actually, why they are different within ecosystems, and what's the different contributing factors for the different organisms actually have not been studied yet. So I think in the future, we need to do some, very very good computer experiment to separate the plants and the microbial, and also the plant respiration. We can also separate from the above ground and the root respiration to individually to explore their optimum temperature and their contributing factors. Yeah, and also when we put them together at the ecosystem levels, whether the observed optimal temperature of ecosystem respiration is more closely to plants optimal temperature or more closely to microbial respiration optimal temperature, all those questions actually have not been answered yet. Yeah, we need to do that in the future. We also cover this in our review paper. So do you have any suggestion how to conduct those kind of manipulative experiment to address your question?
I don't think I can have too much and the suggestion because your lab is so busy and so productive, and so I'm just trying to follow. Okay, and but allow me. Probably one is a push question and one is a back question. Push question is that really valuable even to really focus on the ecosystem thermal optimal? Because the argument here is, are we just talking about you just add the summation of different component? Will that be more valuable just explore the mechanism in each of the component while we always want to talk about whole? But if the whole is simply is addition of the part, let's say 3 part, okay, will that be more valuable to just study the part instead to study the whole? Or argue, if you think about study the whole, it's better to study the part, you better have a good reason, right? That's one. Take a little bit back, probably the more and the testable hypothesis is if we just compare the two major components and the plant and the soil, I think you likely will have the answer. And you discuss a different environment. Let's start is some ecosystem is wet, some is more dry. And you're talking about soil moisture and they have a huge impact on the thermal optimal. And and then you can ask a question is, are those kind of temperature difference and affect more on the plant and thermal optimal on its respiration, or more on the soil?
Okay. And for the first question, I think both the component as a whole are valuable to study. You know, in actually, when we talk about the land carbon cycling, actually we use the whole, right? We use the whole to explain the night sink or source. And also, if we want to explain why the ecosystem respiration as a whole to have those kind of the property, we need to go deep by analyzing the different components. So, I think the both these two parts as the different components and also the whole ecosystem respiration are very valuable. And I also, you know that for the leaf respiration and the microbial respiration, it always, you know, measured individually in different sites. But you know, globally, we can have the FluxNet that measures the whole ecosystem respiration, and also, you know, for the most of the land models, actually, it's you know, take the whole ecosystem respiration as a consideration to predict the future the carbon sink source. So that's your first question. Your second one, sorry, I cannot remember clearly. You know, I just take some flu medicine. My brain is not clear now. So can you repeat? Sorry about that.
The second question is how's the different and the moisture condition will affect the plant part of the thermal optimal versus the soil part of the thermal optimal?
Yeah, good question. We just found this from the FluxNet synthesis, but actually for the plants and the soil, on microbial respiration, how the water impact the optimum temperature. So we have not have any evidence yet, but from our previous studies, on the soil water content impact on soil respiration, I would speculate that it had similar results with this GPP optimum temperature changes. Which means that actually in the dry areas, because of water limitation, the microbial cannot, adjust their thermal activities well. So in dry areas, the optimum temperature of soil respiration or microbial respiration may not acclimate to the temperature well compared with wet conditions. So that might just my speculation. We need some field evidence in the future. Now you are experts, so can you get us from your studies?
Okay, good. So more questions?
Actually, maybe I will ask some
Hi,
questions.
Jin.
Yeah, great to see you. So
Good to
sounds
see
like
you.
you are sick again. Sorry to hear you take yeah taking medicines. So yeah, great talk. I really enjoy it. So I guess yeah I do have some questions related to the idea of the thermal optimal like idea. So yes, I mean, just building on like Wei-Xing's great questions. Maybe first is more technical questions. For your respiration calculation, right? So how do you? I mean, most of the tower will measure NE. You have a daytime, nighttime method to partition that into respiration. So I mean, how did you did the partition, and did you actually consider the temperature optimal already in the partition, right? So does that actually affect your results?
Yeah, very critical question. Yes, we considered that issue. Yeah, actually, NE is the directly measured variables, and actually GPP and ER are also calculated. So to avoid any collinearity between the GPP and ER, actually we use the daytime partitioning method. You know, in the daytime partitioning method, that equation, the GPP and ER use independent modules. So not the ER not calculated by using the GPP minus NEP, not because of that. So relatively, in the daytime partition method, GPP and ER are independent. And in other way, we don't think the partition method may influence our conclusions because, for the partitioning, ER is calculated based on actually hourly data within weeks. You know that, right? So actually for our analysis, we just use the yearly data. Yeah, so we don't think our results or our conclusion, is bears by the partition method.
Okay. Okay.
Thank you.
Sounds cool. Yeah. So since you are on this figure, I'm just curious, how do you separate dry and warm, humid and cold? Because to me, the humid part is pretty good. It's a pretty tight line. But for the drier part, you can see there are much more variation there, right? So I'm just curious, yeah, there could be a way to, make that better, either like a more like subgroups or, along the line. Just curious, do you just using a rated index or you use,
Yeah,
what
we
do
know.
you?
Yeah, we noticed that. Actually, for those dry areas, yeah, we don't have a clear answer right now for how to explain those spread variation in the dry areas. But we think that In the dry areas, they probably have some other, the limitations, like the nutrient and also like the, the air, the VPD impact. And so we have not yet to analyze why in those dry areas the dot points spread so widely. So do you
I
have
guess
any thinking?
yeah, no, I just curious. So right now you just use the soil moisture, for example, to separate these two.
Mhm.
Okay.
Oh,
So
yes. Based on the soil moisture, precipitation, actually we use three different categories, variables to identify the dry versus humid areas. So you are expert in hydrology areas. So do you have any speculation on the possible reasons for?
I mean, you
this?
already hit the point. I just feel like could be like VPD, right? Air part. So right now it sounds like you use soil moisture part. Rainfall soil moisture, they both like a liquid part, right? So the demand part could be play a role, but I don't. Yeah, I guess but yeah, it's hard to I just speculate. Maybe you can add additional variable to explain that better, such as the VPD. But but it's cool.
Yeah, thank you for raising this important issue. So yeah, we will further, to explore it.
Yeah,
Thank
thanks. Good to see you. Yeah.
Hi, Shirley.
Hi, 魏老师。
Excellent talk as always. I have
Oh, thank you.
I have a kind of the technical question here. So you have like you studied 200 some of the size, 29 you didn't find the like the decrease, just the mono increase, but you focus on the 100 at the size. So do you have any idea why the 29 size that didn't show the kind of response? Yeah.
Good question. So actually, we addressed this in our paper. For those
Okay.
29 sites, actually 7 sites are from Mediterranean climate. Yeah, so the temperature range less than 3 degrees centigrade. So it's very difficult to get any temperature response patterns from those limited temperature ranges. For the other 22 sites, actually, the GPP did not show any optimal temperature yet. So we can only speculate that for those 22 sites that did not show any optimum temperature of GPP or ER, partly because these sites may located in those cold areas and they still not the vegetation still not catch up the maximum temperature. It's still have a large safety margins to increase their carbon uptake in the future climate warming. Yeah.
Yeah. So for the 183 size, so basically you only like the look at the temperature and the like moisture, that kind of things. So how about the ecosystem? You may have different ecosystems. The forest, grassland, I think, assume you have all the kind of different ecosystem, right? So how the different ecosystem may play some role here or not influence their response?
No significant difference among those vegetation types. Yeah, we use different categories to group our the sites, but actually did not find any significant difference. So that's I think we can see from these figures. This is the ridge regression results, which actually considers the possible collinearity among the variables. Actually, only these two variables, showed the significant impact. And also for those, cannot be quantified, the categories like the vegetative types. We also, we did not show in this figure for those categories, but we did analyze their impact. Actually, no significant impact of vegetative types.
Okay, thank you.
Thank you.
So, Shirley, I think
Hi.
for your group, you did try to use compare the cable model simulated, temporal response of the respiration to the observations. So yeah, you did find that, I mean, there are differences or errors. So I was wondering, so if your group already tried to identify what the causes behind those model versus observations difference.
As I showed in these slides, actually, we just found that the model actually because most of the model use the monotonic increase of equations, so they underestimated the respiration at lower temperature, but overestimated respiration at higher temperature. So I think most likely it's due to the misfunction they used to simulate the respiration.
Yeah, thank you. Any more questions?
Hi, Shuli, this is Jian Yang.
Hi.
Good to see you.
Nice to
It's
see
been a
you.
long time. And I'm not an ecosystem ecologist, and so my question are probably really basic. I wasn't. Dr. Hui has already asked you a similar question, which is like how since there are so many different flux tower size, how that the different kind of vegetation types may play a role. And this is also back to Dr. Drew's question regarding like the contribution between the vegetation and the microbial community or soil, the above ground and the below ground to the overall ecosystem ER. So and also Dr. Wang also showed that we'll ask you the slide that between the warm and then the humid, the dry and the warm and the humid. So to me, I study forests a lot, so I do know that for those kind of conditions, different kind of climate conditions, the forest there will be different kind of forest, and also for example, let's say for the dry and the warm, you probably in some of the extreme situation you will get a shrub more likely than actually the trees, and then or the grass for the dry and the warm. And for the humid, you probably get more like the tree, different kind of tree species in the ecosystem. And so I just wonder, like, back to the Dr. Drew's question, like, well, which component plays a more important role in that overall ER, and how, say, that your student did actually use the Qinghai's experiment to show that the community weighted height is quite a good indicator. So just wonder how that like kind of the different kind of the vegetation across this different size, and also maybe even in different groups, like between the humid and wet climate group versus that dry and warm group, how that actually then plays really the role in the overall ER and its response to that optimality.
Very good question. Yeah, I think actually we call for this these researches in the future research directions in our review paper. Actually, to further analyze the different contributions of different plant functional types, or we call it vegetative types, to the TOPT of ER. We need to individually to analyze, to study their temperature response functions or curves. Like here, we show that actually for the grass and non-grasses, they should cut different optimum temperature for the photosynthesis. So it's likely that between the dry and humid areas, just because of the different plant functional types, and with different optimum temperature of their physiology, the ecosystem respiration optimum temperature get differed. So, but that's the I think we need to do that in the future studies. So far, I have not seen yet to see each components are within ecosystem to see their contributions for the optimum temperature of ecosystem respiration. Yeah. I think it's very likely that different plant functional types have had different optimum temperature. That's is one of the reasons may cause the difference of the TOPT of ER in different ecosystems.
Okay, cool. Yeah, since we already run out of time, and I have to conclude the presentation today. So thanks again to Professor Liu for your nice, very interesting talk, and also to everyone who asked questions today. So yeah, that's the end of this AES ESA talk. We hope you all keep joining us for the next ones and take part in more AES events. Thank you all.
Thank you. Thank you
Thank
all.
you.
Have a nice good
Everyone.
have a nice
Yeah.
year.
Thank you. You
Yeah.
too.
New Year.